joyous amount of time that will be unnamed. And before then, he came to me from doing a master's at Western Washington University. He's going to tell you all about it. I don't have to tell you anything. Just enjoy the beautiful visuals. And Jake is here in front of us.
Hi, everyone. Thank you for being here on Project Week. Today, I'm going to tell you about some of the work that I've done over the past several years that I'm lumping up into understanding and managing climate change responses across ecological scales. So my field is biogeography, climate, and climate change, the intersection of those things. And the reason that I think that this is interesting is because biogeography and climate have had connections for centuries, even since some of the earliest ecologists like Charles Darwin and Alexander von Humboldt. They had all these theories about why doesn't any species live anywhere. And they spent a lot of time looking around the planet and seeing which species live where and theorizing why not further. And one of the main theories that kind of came out of this was that environmental limitations, and particularly temperature, probably set a lot of species ranges. And this has become interesting in the last few decades, since about the 90s, because recently climate change has shifted temperature bands across Earth's surface in pretty predictable ways, which has effectively kind of made Earth one experiment to say, okay, if we thought that temperature zones set species ranges, now temperature zones are shifting. So species ranges should also probably shift. Or species in one place should have experienced changes in their niche space and changes in their performance. And with this has come a lot of research that has been called a globally coherent fingerprint of climate change, these biogeographical changes as temperature bands shift across Earth. Some of these are quite important because they drive things like disease outbreaks for vector species, ticks and mosquitoes, or harvested species or farmed species, what we can eat and what we can use, or tropical species appearing in the temperate zones and temperate species appearing in the Arctic zones, and even increased species interactions as more species live in different places is one predictor of vaccine causing viruses. Oh, sorry, pandemic causing viruses. So this is all quite popular recently. And it's been kind of a fundamental shift in the field of biogeography because it's made biogeography a field of explaining why things are the way they are to predicting how things might one day be, and also from predicting to managing. Well, what do we do about it? And so today I'm going to tell you about a few different projects that I have been doing that kind of live in this pathway here. And so the first thing I'm going to tell you about is a small scale project. It starts in a place that is not so much unlike this one, Shannon Point Marine Center, which is a marine center in Washington State. And here I studied larval physiological responses to climate stressors. And I focused on the Olympia oyster, which is the only native oyster species to the American West Coast and was and still is a conservation effort species in Washington State. So I wanted to know how climate change will affect early life success of these oysters, and how can we use predicted changes to guide management strategies and guide the restoration plans occurring in the state. So we focused on 3 climate variables, which are changing in the Salish Sea, where I was located: temperature, salinity, and pH, or ocean acidification. And to make a long story short, I'll be doing a lot of that today. We designed this heat gradient, this multi-gradient interaction tank where we could culture oysters in a bunch of all these circles are little cups where we have oysters. And they're each in different combinations of these 3 variables: temperature, salinity, and pH. And they're sort of acted as possible future oceans. And we could collect oyster data in all of them and build models and see how oysters might live in the future. In reality, it looked more like this: a bunch of cups and tubes with little larvae in them. So then in each cup, we took samples of larvae every few days, and we could measure different things like how much they grew, how many of them died, how many of them reached competence, or like they were done being a larva, they were ready to be an adult. And then we could arrange all of these cups by these 3 interacting gradients: temperature, salinity, and pH. So here, I've arranged the 50 treatment cups by temperature going left to right. Oh, no, sorry, bottom to top, and salinity going left to right. And even just by looking, we can see that there's this sort of heat map of conditions that oysters liked. We could also see which ones of them lived and died. These green ones were the ones that lived until adulthood. And then we could take data from all these cups and build models to predict within this treatment space how fast oysters would grow and how many of them would live or die. And I won't go into the details, but we built some models. We found that actually these species in this experiment did not really care about ocean acidification. So everything is predicted by temperature, up to down, and salinity left to right. And so on the left, we have a growth rate model. Those species, the oysters in red treatments will grow faster, blue treatments will grow slower. And on the right, we have what proportion of them we think will survive to adulthood. We have 0 in black and 100% in blue. Then we could merge these two models together and have this kind of predictive plane of how fast will oysters grow and how many of them will survive in all these conditions of temperature and salinity that exist in the real world. And this was built from a lab experiment, so we wanted to kind of extrapolate it to the conditions that oysters were experiencing where we were in Washington State, in the Salish Sea. So we focused on a handful of restoration outplant sites that our partner company, Puget Sound Restoration Fund, focused on. And we knew the temperature and the salinity of those sites, and we knew how fast our models said oysters would grow and how they would survive. And so we were able to integrate these plans into their management strategies. The first thing we did was kind of ground this environmental plane with field samples. So we just took samples of temperature and salinity in some bays, and we saw that actually, like, oysters, these bays weren't that suitable for this species. They were kind of in the low end of survivability and in the low-ish end of growth rate. And then we use future projections from the Salish Sea oceanographic model that show what these conditions should be both in the present, 2014, and in the future, 2095. And we know that this model runs a bit cold, but we wanted to get the trend. So the blue dots were the present version, and the red dots are the future version. Sorry, this is kind of dark on here. And so we saw this positive trend. We saw from the present to the future, all the conditions in Washington State should kind of like get better for this species in general. And so these two lessons together kind of taught us both that in Washington State, this species is actually kind of a climate change winner. It was projected to experience faster growth and higher survival in future compared to present conditions. And also that we could integrate these actual models into the restoration plans of the state. So then they could sample different bays and say, okay, they're this likely to survive in this bay and this likely to survive in this bay. Let's focus on this one, which was very cool. So these were the applied lessons that we learned from this work, but there was also kind of fundamental lessons. And one thing that stuck out to me when I was doing this experiment was that we were there on the map near the border of Washington and British Columbia, but the historical range of this species is basically from around BC to around Baja California. And so we found positive projected changes as temperature increases and climate variables change. And it kind of makes sense because where we were, that was kind of like the coldest place that the species lived. So in fact, they were unhappy. They were cold limited, and they might actually benefit from a little bit hotter temperatures. So this is kind of what set me towards my route to the Sunday lab, where I went from larval biology to biogeography and how environmental tolerances determine where species live. So the second project I want to talk about is kind of leveling up a bit. Instead of focusing on individuals, we are focusing on communities. So if we think that, well, let's see. So we moved from Shannon Point Marine Center, where we learned this lesson about, okay, positive increases near the cold end of the range. And we moved over to Shoals Marine Lab in Appledore Island, Maine, where here, this is in a really, really hot climate warming spot. The ocean in the Gulf of Maine is warming faster than 99% of global oceans. And we had this really rich data set of 42 years of intertidal species surveys, much like the ones that y'all probably did in the last couple weeks. And so we could use these things together, this assumption that increase in time means also increase in temperature, and this rich data set to ask, do population and community level changes align with climate-based expectations? So what I mean by that is, for example, if we're there in Appledore Island, Maine, species that kind of it's the coldest place that they live, they might increase as climate change happens. Species where Appledore is kind of the warmest place they live, they might decrease in performance or abundance as climate change happens. And species that are like kind of well suited, we don't really know what will happen with them. They're harder to predict. And also, we thought that the whole community might sort of do this shift, do this balance, where cold affinity species might go down in abundance and warm affinity species might go up as climate change makes the environment differently suitable for all the species that inhabit it. So I'll tell you about, okay, on the left here, we've got the raw data that we used. All we had was density surveys, people who went out to the intertidal and they counted individual species, snails and starfish and urchins and stuff, and they said 2 urchins per meter. And then we had percent cover surveys of things like algae and barnacles and mussels. And we converted those into both presence and absences across intertidal heights for 42 years and of slopes of abundance change. We could see how the abundance of each species changed through time. And then we don't actually know where a lot of these species live. We don't have this data for marine invertebrates often. So we inferred it by their global occurrences. We took every single species in our data set, about 90 species, and we searched for every single place they've ever been seen on Earth. And then we took the temperatures from all of those places and the coldest temperature of the year, the hottest temperature of the year, and the mean, and we built these thermal affinities. Basically, we know that this species likes zones that are this temperature. And we wanted to see if we could use this information to ask about changes occurring on Appledore Island. So here I'm going to focus on 3 questions that we asked. 1, does relative thermal affinity predict species occurrences? So what I mean here is like species that like hotter places than Appledore Island, they should start liking it more. They should appear in the community as it gets hotter. Species that like cold places, they should probably disappear. Similarly, does relative thermal affinity predict abundance change? So are hot-loving species getting more common, cold-loving species getting less common? And third, does overall community thermal affinity track changes in temperature? So this first question about occurrences, this is where we guessed that if we saw changes in who lives in Appledore Island, we would be able to detect a difference in the thermal affinity of the arriving group and the disappearing group. So here we have all the species in our survey, 90 or so, and we grouped our 42 years into 4-ish year bins. And we sorted the species into groups by their occurrences. Here they're ordered by warm-loving species on top and cold-loving species on bottom. And just by looking, we can see a lot of the pink group, a lot of the arriving species, species that were not on Appledore Island at the beginning of the survey period, but were by the end, are the warmer affinity ones. And the disappearing species, the one that were on Appledore Island at the start of the survey period, but not by the end, are colder affinity. They're towards the bottom. Then we tested for differences between these groups, and I won't really waste a bunch of time on it, but we found that indeed, the arriving species had, in general, hotter thermal affinities than the disappearing species. So species that came in were generally hot species. Species that left were generally cold species, which is consistent with assumptions that we have or past research that has been done on climate change and range shifts and all these things. The second question was, does relative thermal affinity also predict abundance change? So here we modeled the coefficients of all the abundance models. So we had a coefficient of every single species over time. If it's a positive number above the dotted line, it's increasing in abundance. And a negative number below the dotted line means it's getting less common on Appledore Island. And so if we were correct about how temperature drives community change, we should see positive relationships in both data sets. They're separate because of stat stuff. You can't, it's hard. We did different models on each group here. And we found that indeed, in both groups, we found this significant positive relationship. Species that prefer hotter temperatures increased in abundance. Species that prefer colder temperatures decreased. And we also found that these little gray boxes in the middle, that's the true experienced temperature on Appledore Island throughout the sampling span. So the left is the start and the right is the end I don't have a mouse. Anyway, so we found that a lot of species had their thermal affinity kind of within this range. They were actually pretty well suited to live here. And those species particularly, there's a wide belt. Some of them increased and decreased in abundance a lot, more than our models predicted they would. So we think that that's due to all the other parts of ecology, species interactions and changes from entering species, disrupting the environment, all these kinds of things. So we can't predict everything using temperature alone, but we found this positive relationship in both groups. Third, we wanted to know if overall community thermal affinity tracks temperature change. So we wanted to know, as a whole, is the community shifting towards warmer, loving species? So we use this metric called the community thermal index, which is a weighted mean, basically, of all the thermal affinities of all the species in the community. And again, we had to do this separately for density species and percent cover species because they're hard to combine statistically. But in both groups, we found this significant increase in the thermal affinity of the whole community. However, this dotted, oh, you can't really, yeah, you can kind of see it. The dotted line here is the true increase in temperature. So we found that even though the community is shifting towards warmer, loving species, it's not doing it as quickly as climate is actually changing. It's about a 6.5 times slower increase in thermal affinity of the community versus true temperature change. So this is kind of like a climate lag. It indicates that this community might become in thermal risk if climate keeps changing really, really fast and it keeps responding quite slowly. And so this is actually not all we did. We really used these data to address 7 different hypotheses about how thermal affinities and temperatures and climate change should affect species. I don't have time to go over them all, but we found support for 6 of our 7. The only one we didn't being vertical shifts throughout the intertidal zone. We thought things would shift deeper into the water. We didn't really find that. But overall, from this project, we found 6 of 7 globally informed climate response hypotheses to be supported in this climate warming hotspot. We found strong evidence for climate-driven biological reorganization of the intertidal community. We found that community-level variables like richness and community thermal affinity can actually be more sensitive indicators than individual species level metrics, like changes in the absolute distribution limits of the range. Basically, we didn't find things going down in tidal height, but we did find richness going down, things like that. And finally, we found evidence. We kind of inferred that if we're in one spot and climate envelopes in the world are shifting past us and we're seeing changes in the community in our one spot, that's probably evidence that species are shifting their ranges towards the poles, passing us on their way. So that brings me to our third project that we'll talk about, which is a few projects, all about species range shifts. And so this is the idea that has been published on many, many times since about the 90s, that as thermal envelopes shift, species will also shift. And species will track thermal niches as they move across space. And so we wanted to know, globally, if we look at every range shift that's ever been published, are they consistent with what we would expect based on climate change? And second, are they predictable? Can we predict how ranges will be shifting in places that we haven't actually observed them? So the first thing we did when I first got to Jen's lab, basically, is did a huge review on this topic. We pulled together every single range shift observation that we could find in the scientific literature, pulled together over 30,000 observations of range shifts that have been documented. We found, as to be expected, pretty extreme taxonomic and geographical biases in where we've seen range shifts. Here, this is the taxonomic, the taxa, where we've observed them shifting their ranges. It's mostly plants, birds, and arthropods. We have really few data on other species like amphibians and fish and all these other things. And here are the continents in which we have found range shifts. Each bubble here is one study that documented a bunch of range shifts. So there's studies from all 7 continents, but very biased towards North America and Europe. But overall, if we take all of the range shifts that have been documented, we find that about 59% of them are directionally consistent with expectations based on climate change. In other words, they're either upslope in mountains towards colder mountaintops or towards the poles. And this changes a little depending on like what edge of the range you're on and what ecosystem, but the grand total, 59%, which is not that high. And so we started to think of reasons why ranges might not be shifting in the directions that we think they should because of climate change. And I won't get too into this, but we came up with a big list of other drivers that might be influencing how species ranges can move. This is an example of a temperature-driven shift. So this, I wish I had a mouse. This solid line would be a species old range, and then all the red populations might die as climate change shifts that way, and then the blue populations might appear. This is like kind of in a theoretical world what a range shift might look like, going both higher in latitude and higher in elevation in the mountains. But the real world is quite messy. Lots of different things might affect these things. There might be cities in the way. There might be dispersal limitations like wind patterns or ocean currents. There might be microclimates driven by a bunch high precipitation, and it actually keeps things colder. And so this might be kind of why we're not seeing ranges as we think they should go. But we also, something I'm going to focus on a lot and have been focusing on a lot is this little magnifying glass right here on detection methods. It's about whether ranges actually might be shifting in the ways that we're expected to, but we're actually having a hard time seeing it. So one big takeaway from this global review of species range shifts was that we need some synthetic tools. We need some people to pull together data and build us research tools so we can study these things on large global scales. So then we got to doing just that. I worked with this group in France, the BioShifts working group, and we worked to build tools to facilitate biogeographic research. So the way that we did this was we took two big existing databases of species range shifts. One was ours, one was our collaborators. We merged them together, we cleaned them, we made sure all the records match, all these things. Then we got to harmonize the biggest range shift database to ever exist. Then we calculated all these other variables that we think might be important in the detections. We calculated how fast is climate shifting in that region of that study where they saw that species range shift, what kind of methods did they use, what kind of species is it, what's the geographic layout of both the study area where we were looking for range shifts and the species range, up about 16 different metadata variables. And then we wrapped it all up into a software package and made it available to use. So we've been using this package to kind of ask our own questions about range shifts and, oops, sorry, I hit the microphone, and climate change at a global scale. So the BioShifter, this R package that we developed, contains, sorry. Okay, BioShifter contains about 32,000 range shift estimates of over 12,000 species, shifting both across latitudes and across elevations everywhere on the globe. It contains 12 climate prediction variables that we calculated ourselves, and 16 methodological variables that might affect how well shifts are detected. We also collated geographical information and 11 custom functions to manipulate and access the database. It's available, so go look it up if you would like to do range shift research. And inside of this, here's basically an overview of what we have. On the top panel, we have all the range shifts that are found on land across latitudes. In the middle panel is all the ones that are found in the ocean across latitudes. And the bottom one is range shifts up mountains. So in general, again, they're very taxonomically and geographically biased, but generally have slightly positive above zero rates. And so we wanted to take all of these range shifts and all of the extra data that we collected and all of the climate predictor variables and ask, okay, given all of this information, do species ranges actually track climate change? So this working group, we basically took all the ranges, we collected everything that we had, and we wanted to plot range shift rate on the y-axis as a function of species-specific climate velocity. So how fast was the climate moving in the area that that species was when it was seen? So in a perfect world, if we're correct and species are perfectly tracking their thermal niches, we should produce a one-to-one relationship, which is that dotted line. They should perfectly track climate change. And what we found was, drumroll please, not that at all. We found almost no relationship between climate velocity and range shift rate using all the global observations that we had, 17,000 of them-ish, of 15,000. We did find a significantly positive correlation between these values, but the slope of the coefficient was 0.05, meaning that if this is correct, range shifts are happening about 20 times more slowly than climate is moving. So this is actually not uncommon in many studies who have looked at whether or not climate velocity predicts range shifts. A lot of them find the answer's kind of not always yes, or weak coefficients, or coefficients that aren't one-to-one. And we kind of thought a lot while we were collating all these ranges and playing with all these geographical data about why this is. And we thought maybe it's because we're not looking quite in enough places. So before we get to our research on this, I want to remind you of the story of the blindfolded people and the elephant, which is a story from the Indian subcontinent featuring 6 blind men, 6 blindfolded people, encountering an elephant. And they have to work together to figure out what the elephant is. And so each of them is accessing only a small part of the elephant and coming to a different conclusion about what is an elephant. The one who's touching its tusk says an elephant is like a spear. The one who's touching its tail says an elephant is like a rope. The one who's touching its side says an elephant is like a wall. And while each of these things might be true to these people's experiences, they don't represent the full picture of what is happening overall. So we thought that this might be happening when we are detecting range shifts. And I'll explain how. For example, all range shifts that we have ever detected, they need to necessarily be detected within a limited area where you have data. You never have data everywhere. And we're not sure if we have data from this small place, but the species lives in this big place. We're only seeing part of the elephant. Would we even be expected to detect a range shift accurately? For example, maybe we collate all the data on birds in California over a bunch of time. And that's a pretty big space to collate a bunch of data through time. But maybe we detect 400 species of birds. Some of them live fully in California. Some of them have really big ranges, and we're only seeing a small chunk. For example, this one, the American goshawk, lives in all this gray area. And if we were detecting a range shift of this species in California, we would still find something with our data, but would it actually be expected to represent what's happening in the greater distribution of the range? We think probably no. And we think this for 2 reasons. First is that if this species lives all the way from Alaska to Mexico, if we're assuming that temperature drives range shifts, then it's probably not limited by temperature in California anyway. It lives in both hotter places and colder places. So it's unlikely to move at exactly the same rate. And second, because when we're truncating a big data set to a small data set, we're going to have some statistical limitations. We're basically creating a distribution of where this species lives that's only a subset of the true distribution where the species lives, and we won't be seeing that. And so we wanted to see how prevalent this is in all the range shifts that have been collected of these 17,000 or whatever that we collected across latitudes. So we took every single range shift that we have found, documented, and we could find the species range for or build the species range for. We cropped them to the same longitudinal extent, and then we found how far is the species range, north and south, and then how far is the study area, the purple part, north and south. And for example, here, our study area covers about 30% of the species' latitudinal breadth, and it actually doesn't even contain any of the 3 parameters where we usually detect range shifts: the leading edge, the trailing edge, or the range center. So we did this for every single bioshift species. Oh yeah, it's a spear, is what I think that this is seeing. We did this for every single bioshift species that we could, and we found that actually most of the studies that have been done only cover just a little bit of the range. So here, these are range shifts on the top that are focusing on the leading edge of the species range, range shifts detections that are focusing on the range center of the species range, and range shift detections focusing on the trailing edge, or the equator word, the hot edge of the species range. And in each panel, every single gray line is a species latitudinal breadth, and the purple is the study area that detects that species. So we can see that a lot of the purple is quite small compared to a lot of the gray. We're only seeing little bits of the species ranges. Actually finding that less than half of the latitudinal breadth of the species range is covered in most of bioshift studies, and only 26% of them cover at least half. So we thought this might be influencing why our range shift detections don't really make sense. We can't predict them. And I made a little diagram of why we think not here. So here's an example of a species, the gray dots, who lives across latitudes. And maybe it's range shifting, but we are only looking in that purple area. So the gray distribution on the left is truly where the species lives, and the purple on the right is where we're seeing it. And if it's shifting through time, it might actually have a range shift that is happening, the purple dots here on the right graph, but we're not detecting it inside of our little distributional area. The purple, oh sorry, yeah, the gray on the right is the real shift. The purple on the right is what we see. And so we think that this was probably happening in bioshift's data. So we use simulations to get at this. The hard part about assessing 17,000 empirical observations is we actually don't know what's happening in the real world. So we can't actually know if our observations of it are correct or not. But in a simulated environment, we can know. So basically what we did is we simulated a species range shift in some unidimensional space, a y-axis. So here's the distribution of a species from time 1 on the left to time 2 on the right. And then we observed it in a little purple study area, and we detected how much it shifted within the little bit that we see. And then we grew the size of that purple area and saw how the detections changed as our study area increased in size. So basically modeling seeing only a bit of the elephant to seeing the full elephant. And then we did this a bunch more times with study areas centered at different parts of the species range, some at the very north end, some at the very south end. And we quantified all the stuff that we found, all the detections that we made. And then we repeated this with 7 other potential distributions of species, 6 others, because we're not really sure if species are normally distributed across space. And we did this also with species with different levels of detectability, species that are hard to see, these kinds of things. We did a bunch of different simulation parameters, totaling, actually this is outdated. It says 67,000 simulations. Now we have more like 6 million simulated detections of species range shifts. So we wanted to take all of those 6 million simulated detections where we know the true answer. We know how much the ranges were shifting because I programmed it. And what we found based on little places that we looked, where we looked. And so I won't go too much into assessing the simulations, but we kind of came up with 4 takeaways from our simulated range shifts. Is that first, to properly detect a range shift that is happening, your study area, the little purple window, it has to be in the right place. So for example, here is a leading edge, that's the top part of the range, shift that is a true shift of 10 units. And all of these little boxes spread throughout the latitudinal distribution of the range detect different things. The one on the top pretty much detects 10, but the ones in the middle and the bottom and the really bottom detect a lot worse range shifts compared to the true one. Second, your study window needs to be big. Here is a graph of one detection, again, a 10 unit range shift, where we're detecting it with a bunch of little windows on the left and then a bunch of big windows on the right. So the bigger your window gets, the more often we're seeing a detection that represents the true value. Third, you need to be both big and in the right place. So here, again, from left to right, we have small window to big window, or little part of the elephant to whole elephant. And the colors represent where that's positioned within the range. So here we're detecting a leading edge shift, the top part. And we see that the red box, it actually doesn't need to be as big as the purple boxes need to be before it starts seeing a good shift. This is the proportion of shifts that are like kind of right. And finally, we need to have continuous domain. So a lot of species and range shift detections are made in places like the UK, where maybe the species range and the study area both end just at the top of the islands, and then there's no more land. So in those cases, we don't expect to see a range shift because the range can't actually move. It's bound by geography. And so we decided to apply these. We decided that study areas with high coverage of species ranges that are also positioned near their focal parameter should yield the best detections. And luckily, we have all this information inside of BioShifter, our package. So we took our almost 15,000 species ranges and we broke them up into categories. So we did this kind of cumulative filtering exercise on the 17,000 range shifts that we focused on, where the first group was just all of the detections. That's the model I already showed you that was quite bad. The second group was those that contain the focal parameter. They contain the leading edge if they're trying to detect a leading edge shift. They contain the range center if they're trying to detect a center shift or the trailing edge. That was about 46% of all the detections. Third is study areas that both contain the focal parameter and also at least 50% of the range latitudinal breadth. Fourth is study areas that contain the full latitudinal breadth of the species. And fifth is study areas that contain the full breadth and also go a little further. So we know there's a little buffer. We know they're not both at the edge of a continent or something. And the most filtered group is only 5% of all of the data that we had. So about 720 observations of range shifts. So this is basically going from seeing none of the elephant to seeing the whole elephant. We should get better detections as we go across these groups. So we did the same model that I showed you earlier, climate velocity on the x-axis, how fast climate is moving, plotted against the shift, how fast the species is shifting. So if we're correct about this, as we go from left to right, the line should get closer and closer to 1-to-1. I already showed you this one, which is no line. Oh, on the right here, that's just the coefficient. So the theoretical expectation is this dotted line on 1. So with all detections, we get a 0.05 coefficient, not predictive, not good. And then as we go across these groups, we get a little closer to 1-to-1 until our most filtered group has a coefficient of about 0.88, very close to our theoretical expectation of 1. But we weren't so sure about what parameters we should include in our model to be conservative, so we also did this with 20 other models, and we found kind of the same thing each time. Where using this most filtered group, where we think we can see the whole elephant, range shifts actually kind of make sense. They match the climate-based predictions that we made. So that brings us to the elephant in the room, is what do we do with then all of these detections that are made in small study windows, 15,000 of them. And so we basically propose that within a small window, we're not really going to be able to detect the rate of a range shift. But another thing you can get from these same exact data is the total abundance within your window. And if the species is shifting through your study window, you should be able to see changes in abundance and relate them to where you think the species lives and infer what direction the species is moving. And this actually isn't a new idea. People have been doing this for a long time, since one of the very first ever range shift papers published in 1995, where they took abundance changes of all these species in the intertidal in California and saw that southern affinity species increased in abundance in their spot and northern affinity species decreased. So abundance changes actually might be better at predicting the direction of range shifts than trying to measure range shifts themselves. Also with our simulations, we found the same thing. We found when you know where your study area is within a species range, you can probably infer the answer. So the top panel is things near the top of the range. We should always have blue. And the bottom panel is things near the end of the range. We should always have red, abundance decreases that indicate, oh, the species is going away from me now. And we found that especially when the study area is small, on the left side of these plots here, it does a pretty good job. And we know I already showed you this plot, but we know where we've detected range shifts, where we are in the species range, and what proportion that we're detecting. So all of these range shifts that we think are unreliable to actually measure the rate of range shifts might be able to do so by assessing changes in abundance instead. So overall, in this range shift portion, we found that range shift estimates do match climate expectations, given that they are looking in a suitable study window. We found that abundance changes might actually better indicate range shifts than range shifts themselves. And we found that lots of detected range shift rates are probably unreliable in large-scale models. And so last, I'm going to kind of breeze through this chapter. The last kind of portion of my research program focuses on how do we use predicted changes based on climate change to cater management strategies for a changing world. So basically, we wanted to ask, given all these lessons that we've learned, for example, 1, species in certain places will have different reactions to climate change. 2, in Maine, climate change drives community assembly. We know that as places get warmer or colder, the community will also change. And 3, that range shifts do appear to track changes in temperature when you assess them correctly. How do we use all of this to guide our management strategies? We wanted to know specifically, can we direct policy actions to cater to expected ecological changes in fixed locations? So what I mean by this is, for example, now we have a species range, that top thing, all the blobs distributed from the hot end in the red to the cold end in the blue. Maybe it has management areas, these purple boxes. Oh yeah, sorry, we're switching the definition of purple boxes now. Now these purple boxes are management areas. And so what do we do at different edges of the range? Maybe catering our management strategies might be putting new protected areas in specific places that we think species might be going. And then on the bottom, if we have a bunch of species, all these lines is now a species, distributed from the hot edge of the range to their cold edge of the range, we might be able to predict the overall change in different management areas depending on what portions of species ranges they have. If they have a lot of species going away, they might have a different climate outlook than if they have a lot of species coming in, or a mix, a big change of species. So can we kind of cater the management strategies of fixed locations to ecological changes that are predicted? So I won't really get into a lot of this, but we focused on marine ecosystems in Canada, because Canada has a very large and diverse marine environment. It's got the seventh biggest ocean zone in the world, spanning 3 different oceans, the Arctic, Atlantic, and Pacific, and each come with their own changes. And we thought maybe a one-size-fits-all conservation strategy is not the most appropriate when we know different places will experience different changes. And here, this is all the protected areas in Canada, all these purple and orange and pink shapes. And so we worked with partners at University of Miami who have built habitat suitability models for a bunch of species using global distributions and fancy machine learning models and all these things. And we took the habitat suitability models for about 1,200 species of fish that live in Canada, and we projected where they should live in Canada, both in the present, 2010, and in the future, 2100. Then we assessed all the changes across space and asked how is Canada's ocean changing as a whole, and how is communities changing within management regions, and also what can we do about it. And so to make a really long story short, again, we found that Canada's 3 oceans, the Arctic, Atlantic, and Pacific, they're expected to change in different ways. We basically took predictions of all of the species distributions of all of these fish and calculated richness change and community change and how many new species are going to show up in this protected area and how many old species are going to leave and a bunch of things. And we found that there's kind of regional grouping. You can see all the dots of one color represent the 3 oceans. And so they kind of have different patterns here and there. And we won't spend too long looking at this, but we found overall marine protected areas in the Pacific, they're not expected to change much. They have high species richness, both in the present and in the future, and they have very little community change. They're not going to expected to change much due to climate warming. The Atlantic, however, is quite dynamic. It has a big range of changes that are expected. A lot of new species projected to come in, a lot of species projected to leave. And finally, the Arctic is projected to change quite a lot, mostly in relative change, mostly in, there's not that as many species there right now. And so they're going to get a lot of new species, and at least do according to these projections, which might change their community compositions. And so given all of this regional data that we have and specific data, every point here represents an individual marine protected area where there's a total of about 600 in Canada. We thought that each situation requires its own conservation recipe. And so we're working on creating a cookbook. So we did this first regionally, where we took all the Pacific MPAs where in general we can say they're projected for low change. And so our recommendation is here, we have to design monitoring strategies that are powerful, statistically powerful, where we can precisely track changes that are small apart from random variation. And so myself and Jen and Eden Tekwa have been developing a power analysis for biodiversity sampling to do so, to say how do we get the best detections for the lowest cost and the most efficiency in this region. Second is Arctic. They are expected to gain lots of species. And so here we think that we need to codify plans for managing newly arrived species in environments. So we're working with colleagues at the UDES Geological Survey on adapting the RAD framework, which is the Resist, Accept, Direct framework for range shifting species. So this means every species we think might enter, what do we want to do with it? Do we want to keep it away? Do we want to help it come in? Or do we want to do nothing and just let whatever happens happen? And finally, the Atlantic MPAs are characterized by high turnover and high dynamicity. Just different things are going to happen in the Atlantic. It's changing a lot. So here we suggest research into basically the impacts of incoming or outgoing species. Might we have any ecological cascades? Might we lose any important keystone species? That kind of thing. And so we actually partnered with the Climate Change Committee of Quebec for this project, who had requested from us specific recommendations on policy guidelines for climate change. So we gave them these regional guidelines and also site-specific recommendations for every single marine protected area in Canada. We're not gonna, it's kind of the same things, based on their projected changes, how many species they have, how many fishery species they have, etc. And second, yes, okay, sorry, I skipped a slide on. We also helped identify new places to put MPAs to kind of catch range-shifting species, which is the Gulf of St. Lawrence in Quebec and kind of the Arctic. So overall, we found that yes, we can use projections to help build kind of climate-wise conservation strategies in Canada and probably the world. So overall, my research aims to both detect, predict, and manage climate-driven change at multiple scales, from individuals to communities, create synthetic computational tools, and suggest guidelines for conservation management. And that is all I have to talk about. Thank you for listening.